Hello students, this is the review video for Unit 5 Molecular Genetics. Alright, so we're going to start out, if you remember, the central dogma of molecular biology is the flow of information from DNA to RNA to proteins. So in that, there's two processes that occur. There's transcription, which is being used where we use DNA to make RNA, and then there's translation, where we use RNA to make that chain of amino acids. And we'll talk a little more specific about how each of these processes happens here in just a moment. So transcription, DNA to RNA, happens in the nucleus, and then translation, going from RNA to protein, actually happens in the ribosome. So let's talk about RNA and DNA and talk about some differences. So RNA is basically your link between your DNA and protein. It stands for ribonucleic acid. So it's the nucleic acid specifically used to make proteins. So in DNA, if you remember, it uses the sugar deoxyribose. RNA uses the sugar ribose. RNA is single-stranded and sometimes can be twisted, but it is different because DNA is double-stranded or has that double helix. And then as far as our nitrogenous bases, in DNA we have A, C, G, and T, but in RNA we use A, C, G, and U. So we use uracil instead of thymine. All right, so let's talk specifically about transcription. So transcription is going from DNA to RNA. So our two strands are going to separate, and that's where transcription will take place. RNA polymerase is then the enzyme that allows transcription to actually happen. So it's the one responsible for linking those nucleotides together in the strand of RNA. All right, so let's review translation. So if you remember translation, we talked about codons. So translation is going from the strand of RNA to our chain of amino acids. But we have to talk about codons because codons are those individual nucleotides, or those group of 3 nucleotides, and those 3 nucleotides together make up a codon. Okay? So remember, we had stop codons and we had start codons. There's only one start codon, which tells that strand of amino acid where it needs to start as far as the translation process to make those amino acids. And then the stop codons tell it essentially when to stop. So remember, this is your amino acid table. These are abbreviations for those amino acids. And then again, so translation is going from RNA to your chain of amino acids, or RNA to protein, and this whole process happens in the ribosome. All right, let's talk about mutations. So if you remember, a mutation is a change in an organism's DNA. So it happens during that transcription, or can happen during that replication process. So mutations may or may not affect your physical traits. So sometimes it'll affect the phenotype and sometimes it will not. We're going to talk about different types of mutations. So there's single gene mutations, which are point mutations and frameshift mutations, or there's larger chromosomal mutations. So single gene mutations, you have a point mutation, which literally just replaces one nucleotide with another. So if you have ACGT, it's going to then replace maybe that G with a C, so it'd be ACCT instead. A frameshift mutation is actually one that is going to shift that reading frame. So it's inserting either an extra nucleotide, or it can delete an actual nucleotide out. Frameshift mutations tend to have greater ramifications than a point mutation. All right, then there's chromosomal mutations. So these are mutations that affect the entire chromosome. Okay, so these typically occur during meiosis, so the first part of meiosis during crossing over. So you have gene duplication when a part of that gene is repeated in that chromosome, so it literally is duplicated. And then you have gene insertion, so you can have the insertion from a piece of a chromosome from another chromosome inserted into another chromosome. Gene deletion, where a gene is actually removed from that chromosome. Gene inversion, where the order of genes can actually be switched. And then there's translocation, where you can actually exchange pieces of chromosomes, of non-homologous chromosomes, with one another. And then there's non-disjunction, which this happens when your homologous chromosomes do not separate correctly during meiosis. So this ends up creating errors in your chromosome numbers and your gametes. All right, so again, mutations can have an effect on the phenotype, so it can change your physical traits, but they also might not. Some mutations are considered silent, and they will not have actually any physical effect on the organism or the physical phenotype. All right, and then mutagens. Mutagens are essentially the physical or chemical agents that cause a mutation. So these are things that are actually causing mutations to occur. So these are things outside your body. So a physical mutagen could be like high radiation, and chemical mutagens are things that are similar to normal DNA, but they can cause base pairing to not happen correctly. All right, so now we're going to talk about stem cells. So if you remember, stem cells are those cells that remain undifferentiated. Basically, they are cells that are able to change into any different type of cell. So if you remember, your adult stem cells are able to turn into certain specific things, but are not able to differentiate near as much as your embryonic stem cells. So in your body, your cells normally differentiate into specific types. So you can have a skin cell, blood cell, muscle cell, bone cell, nerve cell, all these different types of cells. And stem cells are ones that are able to differentiate into all those different types of cells. And this happens by your cells only expressing certain parts. So certain parts of the DNA that are inside those cells are actually transcribed and translated to determine what the function of that cell is going to be. Okay, so now we're going to talk about manipulating DNA and genetic engineering. So let's talk about restriction enzymes. So if you remember restriction enzymes, those are those tools that are used to cut DNA. So it actually cuts the bonds between nucleotides and allows for those fragments to be separated. So we can use restriction enzymes. We use restriction enzymes when we want to do gel electrophoresis. So remember, that's the process of sorting those DNA fragments by length. So if you remember when we did the Who Ate the Cheese activity, we had to, the restriction enzyme recognized a certain sequence and was able to cut those sections of DNA, and then the shorter fragments traveled a much farther distance and the longer fragments traveled a shorter distance, creating this picture, essentially creating an image that looks like this, which is your gel. So you can compare the DNA of one organism to the DNA of another. All right. So then we have to copy DNA. So this process is called polymerase chain reaction, or PCR. So that's a technique. It makes many copies of a certain segment of DNA. So if you were in the crime lab, say, and you didn't have a large enough sample of DNA, you would use this PCR technique in order to make more copies of that same section of DNA that you have in order to perform more tests. All right, so DNA fingerprinting, that's the unique banding that you end up on when you do gel electrophoresis. So if you see each of these lines, these are individual banding that occurs in an individual person. And then no two people will actually share identical repeats in several locations. So that is where you get your DNA fingerprint. All right, and then genetic engineering. So here's where we use some of these different techniques. So there's recombinant DNA, which combines genes from different sources, even different species, into a single DNA molecule. So with recombinant DNA, you're literally taking the DNA of one organism or from one source and injecting it into another source. So you're combining DNA together. So we use plasmids. So this is kind of how this whole process works. So you use a restriction enzyme like we talked about. It cuts that DNA fragment. And then the DNA fragment from the other source will actually be added in. And then the fragments will stick together by those base pairing rules like you see here. And then DNA ligase, that's an enzyme that will paste these ends of those fragments together. And then you'll put this in a bacterial cell, so it'll take up that recombinant plasmid. And then when the bacterial cell divides, it'll create more copies of that plasmid. All right. And the last thing is the Human Genome Project. So if you remember, this was a big project that began in the 90s. So some DNA technology was available, but scientists were trying to work, and ideally they want to identify and map all human genes. And by doing this, it will allow us to compare structures of the human genome to other organisms, allow us to identify previously unknown genes, and also ideally determine all the nucleotides in a person's DNA. That's all we have for our review video. If you have questions, please see your teacher or any of the other